Bismillah Rahman Rahim, 9700 A level biology, Cambridge syllabus, June 2022. This is the new syllabus. So, the new syllabus, this is the first exam that we had, and I'm going to discuss the way how the paper has changed and how the format has changed. How the paper is now for 1 hour 15 minutes, and why? Because it's slightly a more difficult paper. As you can see here, the paper now is for 1 hour and 15 minutes. Previously, it used to be for 1 hour. And this extra time that you've been getting in this paper is because the questions have become slightly more difficult. I mean, I attempted this paper this morning, and it took me at least 2 hours to really do it. Of course, I had a bit of tea in the middle, but it is a little longer, the paper, and it is a little more complex, as I feel. Please bear up with me with my voice because I'm just recovering from another second episode of Corona. COVID-19 infection, and I still have not fully recovered, but I thought I must work on these videos so that I can help you all out in the next coming exam in November. Now, let's look at question number one. The photomicrograph shows a bronchiole and alveoli. So these are the alveoli, all these are the alveoli, and this is the bronchiole. Now, it says the magnification of the image is into 360. What is the maximum diameter of the bronchial lumen? So you've got to find out the diameter. So you have to measure this with a ruler. You have to measure this with a ruler and then say this is, I think, about 5 centimeter. I'm not sure. And so this will be about 50 millimeter. Now 50 millimeter, what have you to convert it into? Because the answer here is all in micrometer. So this will equal to 50,000 micrometer. So always look at the units in the answer. What units do they want in the answer? And then you divide this 50,000 by what? You divide it by this 360. And it really comes to something like the answer is C. So you always divide it with this, the 360 which was given to you, and you convert it first centimeter to millimeter, then millimeter to micrometer. If this answer was they were asking you in nanometer, then you would have to convert everything into nanometers. So always look at the answer. Are the answers in micrometer or the answers in nanometers which they are asking for? Now coming to question number two, a specimen is observed twice with a microscope, firstly using green light with a wavelength of 510 nanometer and using red light with a wavelength of 650 nanometer. What happens to the magnification and resolution when using red light compared to green light? Well, whenever you are using any light, the magnification is going to remain the same. So this was wrong. Magnification is not going to be. Magnification is dependent on the lenses that you have in the lens. I'm sure you've all been studying and doing lab work. It's the lenses X10, X40, whichever one you use, that is the magnification. How is that going to be affected by the wavelength of light? So you knew that A and B was rubbish. Now C and D, now we have to come down to it. Now green is 510 nanometers, so the half the wavelength is 255. Red, it is 325. So what has happened is now two objects which are 325 nanometers apart can be seen clearly. Previously, two objects which are 255 nanometers apart. So what has happened? It has decreased. So this is the answer is C, that it remains the same, it has decreased. Now I'm going to explain this to you, so just hold on a bit because I'm sure this is not very clear to quite a few of the students. What is resolution? Resolution is how detailed the image is, how detailed the image. Don't say it is clear. Clear is a wrong word. It is how well the microscope distinguishes between or separate two points, this point and this point. And if you keep on bringing it closer, a point comes when you can't see them as two separate points that are close together. The higher the resolution, the greater the detail you can see. Just like some pictures, you zoom them and you can't see any detail because they are very bad quality. And that's what actually is on your phones. You have, the megapixel cameras and all that. Now, I want you to also understand another concept is this is from your book. Now you see it is half the wavelength of light. In a light microscope, you can see anything which is less than 200 nanometer because the wavelength of light is 400 nanometer. Now, stained mitochondria of diameter of 1000 nanometer interferes with light waves. Now, stained ribosome of diameter 25 nanometer does not interfere with light waves. So mitochondria and some ribosomes in the path. So if you can see what is going to happen, if there is going to be interference, only then that can be seen. So if something less than 200 nanometer, like ribosomes are less than 200 nanometer, you can't see them in the light microscope. Now I've given you these 2 points like 40 nanometer, 60 nanometer. Now if these 2 points are separate, you can see them separately. But if you bring them closer together, so if the resolution is 40 nanometer or 60 nanometer, this is what you've got to figure out. As the size, as the length between the 2 points decreases, it becomes better, it becomes a higher resolution. So it's sort of difficult to understand that the smaller the distance between the 2 points, that will realize in making it more detail will be seen of the diagram. The third question, four students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by number. One function was produces the mitotic spindle during cell division, so this has to be centriole. And synthesis of polypeptides has to be ribosomes. Synthesis of lipids has to be smooth ER. The appearances were listed by letters. V, membranes which surround an enclosed inner cavity, non-membrane bound spherical structure that looks like ribosomes, a double membrane interspersed with pores, that's nuclear membrane, non-membrane bound cylindrical structures is, the centrioles, which are non-membrane bound, and they are these cylindrical structures, and membrane bound sacs arranged as a flattened stack. Now, this would be the Golgi apparatus. Which student correctly matched the numbered functions with the appearance of the cell structure? Now, A was what? One was produces mitotic spindle. So mitotic spindle would have to be this one. Mitotic spindle would have to be Y. So then immediately I thought of it as Y, and then I looked at Y as one Y anywhere, and then C and D. So I said, okay, now we narrow down. So these two are wrong. This is the important way, is that you first narrow down to one which are two are correct, and then you read the question again and again a number of times. So why? And then it said the second was synthesis of polypeptides. So that has to be ribosome. Now, which one is a ribosome? Ribosome is a non-membrane bound. So that would be W. And three was a synthesis of lipids. So SCR. So that had to be membranes which surround an enclosed inner cavity. You see, this SCR is sort of like this. There are these structures like this. You have to understand. And so it would be. So the answer would be C in this case. SCR, centrioles, and ribosomes. But you have to know the structure to be able to really do this question. Now, coming to question number 4, what is found in chloroplasts and mitochondria? So it has to be present in both. 70S ribosome only, yes, is present in both. 70S ribosome and 80S and circular DNA, yes, that's correct. 80S was wrong. 80S is not present. Circular DNA only, no, because we have the ribosomes as well. So the answer is B. But then what other things do they have in common? I put this diagram here. This diagram was not in the paper. I put it myself. Now basically, what else do they have? They have a double membrane. I'm thinking of a paper which could come in the November exam. Both of them have a double membrane. Both of them, of course, one of them has a double membrane. In the mitochondria, the inner membrane is folded. But in the chloroplast, the outer membrane is there as well. So both of them have a double membrane. And what else do they have in common? Both in both of them, ATP is made. Yeah. So I mean, you could get a question like that. ATP is made in both of them. In one by oxidative phosphorylation and substrate level phosphorylation, and in the other by photophosphorylation. So both of them have ATP. So I mean, we could get a question like that as well in the next coming exam. Now, question number 5: which feature is correct for all known viruses? Capsid made of lipid and protein. Well, we've studied that there's a protein coat, and inside it there can be a DNA strand or there can be an RNA strand. So we have two types of protein. One is the DNA and RNA. So there's a protein coat and the DNA or an RNA strand. So DNA core, I would say this was wrong. Why? Because it can be DNA or RNA. Capsid made of lipid and protein. No protein coat. Outer envelope of phospholipid. No, that's not present in every virus. So non-cellular structure is the only one which makes sense and is the one which is common to all known, because it says feature correct for all known viruses. So basically, they are non-cellular. That's why they're called virus particles, and they're non-cellular or acellular. They do not have a cell like a cell membrane, cytoplasm. They're not even a unit of life. So that is what you have to understand. Now, coming to question number six, four extracts from different plant materials were made and tested with Benedict solution. The extracts were boiled with Benedicts for 240 seconds, and the final color was recorded. So the final color is red, yellow, blue, and green. Well, how I did it was I thought of red. Red is the highest. So is red should be which sequence of plant extracts represents an increasing quantity of reducing sugar. So I thought, red should be right at the end, and there's only one in it. So that's why the answer was D. You see, blue means no reducing sugar, green means slight reducing sugar, yellow means a little more than that, red means the maximum amount of reducing sugar in any sample. Now coming to question number seven, which properties that are dependent on hydrogen bonds? Which have properties that are dependent on hydrogen bonds? Cellulose, does it have properties? Yes, holding the molecules together. Molecule of hemoglobin, yes, would have hydrogen bonds. There's alpha helix and beta pleated sheets, and in water also for its cohesion and adhesion, hydrogen bonds form between the water molecules and the wall of the vessels. So, which have properties that are dependent on hydrogen bonds? So, the answer is A, 1, 2, and 3. Then, question 8, which statement is correct? Now, you have to read all the statements. Cellulose, glycogen, and amylopectin are all polymers. Okay, fine. Well, that is the correct answer. But why is these wrong? You see, why is B wrong? Now, let's look at why B is wrong. Ribose, amylase, and phospholipid are all macromolecules. Phospholipid is a molecule, but ribose is a 5-pentose sugar. It's not a macromolecule. Then starch is not a monomer. This was the mistake in it. Sucrose, deoxyribose are polysaccharides. Sucrose is a disaccharide. It's not a polysaccharide. Deoxyribose is a single 5-carbon sugar. It's a pentose sugar. Amylopectin, yes, is a polysaccharide, but these two are not polysaccharides. So if you didn't know it, is something very that you must know your bio very well to be able to do these questions. They don't just you can't just do them knowing it, sort of knowing your subject very, very pathetically. Now coming to question number 9. The diagram shows two amino acids. Some of the hydrogen atoms are numbered 1 to 6. Which two numbered hydrogen atoms could contribute to the production of a molecule of water when a peptide bond forms between these two amino acids? Now, if you know this very correctly, you're very right. It has to be hydrogen of the amino group here and the OH of the COH. So it can be between 3 and 4, but there is no choice of 3 and 4. So what other choices we have? We have 1 and 4. Well, that's all rubbish. That's wrong. Because 1 is here and this NH is again of the amino group. No, it can't be that. So then B, 1 and 6. Yes, that is possible. It can be between 1 and 6. So that is the answer to this. You see, why are the other wrong? 3 and 5. This is 3. This is the middle hydrogen. ये तो बिल्कुल नहीं हो सकता. This can't happen. 2 and 4. 2 and 4. 2 is again the central carbon atom. So this is also wrong. So these were wrong. But you must know why they are wrong, you see. I mean, I'm not saying that just try to just say, okay, well, this is the answer, so it's correct. But why were the others wrong? That is the most important technique of studying for A-level biology is that in the MCQs, you must know why the others were wrong. Because if you don't know this, then you're going to stuck up with those 10 questions in which you're going to just be figuring out, okay, well, this is not the answer, this is the answer, this is not the answer. So remember this. Whenever you're doing MCQs, look at the answer A, which is correct, and then say, okay, why were the other three wrong? Now coming to question number 10, a student wrote 4 statements about water. Water has a high specific heat capacity, which maintains the temperature of water within cells. Yeah, correct. Mammals rely on water having a relatively low latent heat of vaporization to keep them cool. No, when sweat evaporates, it takes a lot of heat from the body. Otherwise, we would be dehydrated. So, it's a high latent heat of vaporization. So, this is wrong. So, look at the ones in which 2 is wrong. So, these are the ones 2 in which 2 is wrong. When number 3, when a negatively charged ion is added to water, the partial positive charge on the hydrogen is attracted to the ion. Yeah, that's correct. That's correct. When surrounded by water, non-polar molecules tend to be pushed apart. How is that? That's not possible. Non-polar and water, of course, will not react with each other, will pushed apart. So this is wrong. So that is why the answer is B. is that the correct was 1 was correct and 2 and 4 were incorrect. Now coming to question number 11, typical enzymes are large globular proteins with a specific tertiary shape. Which molecular interactions are directly involved in maintaining the tertiary shape? Hydrogen bonding, yes. Disulfide bridges, yes. Hydrophobic interactions, yes. So the answer is A. You see, you have to know is primary structure is only peptide bonds. Secondary structure is hydrogen bonds only. Tertiary has all the four: hydrogen, ionic, disulfide, and hydrophobic interactions. Now, coming to question number 12, which statement by the Michaelis-Menten constant is correct for an enzyme with a low affinity for its substrate? It has a high Km and reaches Vmax at a high substrate concentration. Well, that's correct, but you see, why are they wrong? It has a high Km and at a lower substrate concentration. Now, this is where you didn't know this if you didn't. Low Km, of course, was all wrong because low affinity means high Km. So it's a sort of an inverse relationship that we have with it. Now I'm just going to explain this to you. First of all, what is Vmax and what is Km? Info about maximum rate of reaction that is possible, of course. So this is the Vmax that we see here, right? So enzyme 1, enzyme 2, then we calculate the half Vmax and then we see where it cuts the graph and then we make the, so this is Km1 and Km2. You can see Km1 is less. So smaller Km for enzyme 1 reflects a higher affinity of enzyme. Smaller Km, higher affinity. Larger Km for enzyme 2 reflects a low affinity of enzyme for the substrate. And you can see how the substrate is changing. You see, large Km means more substrate. It is higher, the substrate is increasing this way. So measures the affinity of the enzyme for its substrate. Higher affinity, more likely product will be formed when substrate enters the active site. High affinity produces low Km, low affinity produces high Km. So this is what you have to understand. Another very good graph. I want you to look at it and see how the difference for the normal, then the competitive and the non-competitive inhibitor. So how the Km changes for all these. Question number 13. Long chain saturated fatty acids. Now, saturated fatty acid, long chain, change from solid to liquid at higher temperatures. So naturally, if you heat butter and ghee, butter is solid, but when you heat it, it becomes liquid at higher temperatures. So compared with short chain unsaturated fatty acid. Now, which fatty acids would be more likely to form triglycerides in mammals that live in cold climates? So we want things which will going to remain, they shouldn't solidify. So form triglycerides in mammals that live in. So of course we understand that which fatty acids are more likely to form that live in cold climates. So we want them not to solidify. So that had to be short chain unsaturated. You see, saturated fats are the ones which solidify. Unsaturated, like almond oil, olive oil, they would never solidify. So this is the main thing which they were asking you, that in cold climates, we don't want things to solidify in your cell. Even if your cell membrane solidifies, well, that cell is going to die. So coming to question number 14, when animal cells are cultured, salt solution is added to keep the cells alive. You see, we want animal cells means we want more and more and more of the animal cells. What is the purpose of the salt solution? To allow facilitated diffusion of salts into the cell, to prevent diffusion of other ions into or out of the cell, to prevent net movement of water into or out of the cell, to provide a source of energy for active transport. Now, why is the answer C? The answer is C because you see, if a cell, we have to have no net movement of water. Why? Because the cell should not shrink or it should not burst. So basically, why do we have a salt solution? Salt does not provide energy, so this was wrong. To prevent diffusion of other ions, why would we keep it in a salt solution to keep other ions? This was wrong. Allow facilitated diffusion, why? Why would we want salts into the cell? So the answer is C, and the main reason is to keep the cells alive. So that means they should not either shrink or swell up and burst, because these are animal cells. They don't have any cell wall. They are animal cells. Then coming to question number 15, the following are all processes that allow movement into cells: phagocytosis, active transport, facilitated diffusion. Which process requires ATP? Facilitated diffusion does not require ATP. That's a passive process. It just needs a channel protein, and facilitated diffusion means from 100 to maybe 10 is just going to facilitate the diffusion. Diffusion is not an energy consuming process. It does not require ATP. So which process requires ATP 1 and 2? It was only phagocytosis and active transport. Now coming to question number 16, which features are required to allow for efficient diffusion? A large surface area, yes. A short diffusion pathway, yes. Maintenance of a constant diffusion gradient. These are the points in your book. So the answer is A to this. Large surface area means a cell with this surface area, there's a cell with this surface area, then a short diffusion pathway, the capillaries are here, and they're going to be quick diffusion pathway, and a maintenance of a concentration gradient. It's always 100 outside and 10 inside, and it's continuously being maintained like this. So the diffusion gradient is going to be there all the time. So this is the reason why all these three were correct. Then coming to question number 17, what is the role of mitosis? Now, this is a favorite question, been asked many times before also. Is growth of organisms, production of genetically different cells? That was wrong. And repair of cells, that's I've told you many times, this is a very favorite question. It's not growth and repair of tissues, not repair of cells. A cell can never be repaired. And replacement of cancerous tissue, that's not a role of mitosis. That's just one, mitosis does that, but that's not the role of mitosis. Mitosis has got more healthy roles than having a person developing cancer. So the answer is A, which is the growth of organisms. Then coming to question number 18, telomeres prevent the loss of genes from the ends of chromosomes during DNA replication, but they become shorter at each time they are copied. In cancer cells and stem cells, the telomeres remain the same length. Which statement is correct for all human cells? If telomeres become too short, a cell may stop dividing. Yeah, that's right. That's the answer. But why is B wrong? Adding telomeres could increase the rate of aging cells. No, I mean, how would you add telomeres? Aging is when the telomeres become shortened. Telomeres are repaired by the enzyme RNA polymerase. No, telomeres are at the end of your DNA. So why would we need RNA polymerase here? Telomeres prevent all damage occurring to DNA molecules. No, the rest of the DNA molecule is not affected. The telomere is only the ends of the chromosomes. So the entire DNA is not affected by it. Now coming to question 19, the nucleus of a mouse body cell in G1 phase of the cell has 1.8 into 10 raised to the pulse. So here we have 1.2 into 10 raised to the power minus 12 grams of DNA at this point. 1.2, I'm not writing the whole thing. Now, what will be the mass of DNA in the nucleus of the cell at the end of S phase and at the end of G2 phase? So S phase is what? S phase is when DNA replication takes place. So the 1.2 is going to become the 2.4 because this is when the DNA replication is taking place. And it will remain the same here. So it will remain 2.4 here. So 1.2, so its end of S phase will be 2.4. And at the end of G2 phase, it will also be 2.4. Why? Because DNA replicates only once. This is the S phase in which it replicates, so it will stay 2.4 here till this point. And then, of course, mitosis will take place and all that will happen. So that's why the answer is D. Now, coming to question number 20. What occurs during prophase in animal cells? Fragmentation of the nuclear envelope, yes. Nucleolus disappears, yes. Stained chromosomes become visible. Why? Because they condense and shorten. Centrioles replicate, that's wrong. So that's why the answer was B, 1, 2, and 3 only. Then coming to question number 21, which statement describes the structure of ATP? It is a DNA nucleotide. Well, that's wrong. It's a ribose sugar. So the first two were wrong. RNA nucleotide with two extra phosphates. Yes, that's correct. It's an RNA nucleotide with three extra phosphates. No, a nucleotide already has one phosphate. A nucleotide already has one phosphate in it. A structure of a nucleotide is what? Deoxyribose, one base to which is attached a phosphate. This is a nucleotide. So when you add two more to it, then it becomes ATP. So you just have to add two more to it to make it ATP. So that's why the answer is C and the answer was not D. Okay, then coming on to question number 22. Now coming to question number 22, rifampicin is an antibiotic used to treat tuberculosis. It works by inhibiting RNA polymerase in bacteria. Now RNA polymerase is only used in transcription, DNA to RNA. Which processes are directly inhibited by this antibiotic? Replication, no. Enzyme synthesis, yes, because in enzyme synthesis you'd have to have transcription and translation. ATP synthesis, no. So that is why the answer is D, 2 only, because RNA polymerase is only needed when we have to make mRNA. So that is DNA to mRNA, which is transcription. Please remember, in a bacteria, transcription will occur in the cytoplasm because there is no nucleus. And then another question can come on that as well. So remember that bacteria does not have a nucleus. Then coming to question number 23, the table shows the DNA triplet codes for some amino acids. That's fine, you go through it. The base sequence on the template DNA strand coding for part of a polypeptide. So this is the DNA. Sometimes they'll give you RNA and DNA and then you get very mixed up and then you get it all wrong. Two mutations occur in the sequence during DNA replication. Which mutated template DNA strand would result in a shorter polypeptide? Now, how I did it this morning was I looked at the stop codon because that would make it smaller. Because the stop codon, if it comes early, then of course. So I looked at for ATC anywhere. And in any of these, when I found the ATC, I found it here. So I knew the answer was C. And that's my way of doing it. You might do it in another way, but this is how I figured it out. That there would be a shorter polypeptide would mean that there is some of the amino acids are not going to get in, so it's going to be a shorter number of less number of amino acids. So there had to be a stop codon coming somewhere, which would of course make it shorter. And now coming to question number 24, some of the features present in transport tissues are listed: lignified walls, cytoplasm, mitochondria, pits, plasmodesmata. Which features are present in xylem vessels? Now, of course, there's only lignified walls I could think of, and the cytoplasm is not present, mitochondria is not present, plasmodesmata and pits are present. So the answer was C, which is 1 and 4 only. Now, the diagram shows transverse section through parts of plants. Which row is correct? Contains lignin, of course, that means the xylem. Transports organic solutes, that means that would be the phloem or the sieve tubes. So the answer is B, as I've just labeled it for you: 1, 4, 6, and 2, 3, and 5. Then coming to question number 26, which molecules form hydrogen bonds with water during transpiration? Does cellulose in the xylem form hydrogen bonds with the water? Yes. Adhesion. Suberin, no. Other water molecules in the xylem, yes. Cohesion. So that is why the answer to this was C, 1 and 3 only. Okay, then coming on to the last question of this video, question number 27. Some plant species can take up heavy metal contaminants that are dissolved in soil water and then transport them within the plant. Within plant cells, the heavy metals accumulate mainly in the vacuole. Which suggestions about the transport and accumulation of heavy metals are valid? After initial entry into the root, some of the heavy metals can pass through the tonoplast to be stored in the vacuole of the cells in the core. That makes sense, yes. The heavy metals take an apoplast pathway in the xylem, but at the endodermis they take a symplast. Yes, that seems correct as well. The rate of accumulation of heavy metals in leaf cells will be faster at night when photosynthesis is not occurring than during the day. Why? Now, does it make sense? Why would that happen? The presence of heavy metals may cause transpiration stream causes transpiration. Why would it cause the transpiration stream to slow down? So that is why the answer is A, which is 1 and 2. So now this video finishes here, and I will continue the rest of this paper, and inshallah soon I will be posting the video on online, and this will be posted, and then the next one will follow very soon. Thank you very much for watching, and thank you for subscribing, and thank you for all the good comments, and please, some of you got a very good grade, and I'm glad my videos were helpful. Thank you very much. This is the video 1 on the June 2022 paper.